Hello, Ma'am over here, and welcome to another lecture video for General Biology. In this video or series of videos, we will be covering Module 2, which will discuss about cell types. Now, looking at this slide, I hope you do recognize this. This is one of the pictures taken from our experiment number 1 involving particularly Allium cepa or the plant cell. So as you can see, the cells are large in size. So if you would like to try to make your own observations, the cells are significantly larger in size. They have outlines, visible outlines in dark color, which are the cell wall, a specific detail that is present in plant cells, and the black circles in the picture are the nucleus or the nuclei for each cell. Next, we have over here the animal cell from humans. So this sample is the buccal mucosa slide, also known as the cheek cell. So how is this taken from the person who donated the sample? They simply took a Q-tip or cotton buds and then they lightly scratch it on the inside of their cheek, put it on the slide, put some metal dye, and then there you have it, the sample slide. So comparing to the plant cell that we have viewed in the previous slide, you would notice that for animal cells, the size is also quite large, but the shape is also different. With plant cells have specific shapes or patterns, animal cells have no cell wall, which means their cell membrane is more fluid and they have irregular shapes. The dots or the circular dark areas pointed here by the arrows are the nuclei for each cell. Now, if you are looking at this slide, you would notice that there are multiple rod-shaped bacteria in each cell. So if you would notice the dots, the many dots in each cell, those are actually bacteria, another type of cell. Now, please take a moment to realize that one animal cell is much bigger than a bacterial cell. So in other words, one animal cell is much bigger in size, while bacterial cells are typically very small. And that's what we're going to discuss today, like the variations, similarities, and differences between the different types of cells, namely prokaryotes, animal cells, and plant cells. Alright, so here is the title of our module, Cell Types and Modification. For our learning objectives, they are as follows. Number 1, we will be distinguishing prokaryote and eukaryotic cells according to their features. So from the previous discussion of Module 1, eukaryotic cells have been discussed in detail. So we gave the organelles. Now we're going to discuss the difference, the signature or main characteristics of prokaryotic cells. Next, we have classified different cell types, namely plants, animal tissues, and specify the functions of each. Next, we will also describe cell modifications that lead to adaptations. So as we continue, we will be discussing like unique characteristics that are present for prokaryotes, eukaryotes, plants, and animals. Alright, so in all of life forms here on our planet, you can classify them into two groups based on the presence of their nucleus or the absence of their nucleus. These two groups or classes are the prokaryotes and the eukaryotes. So the members of the prokaryotic are only bacteria. So please bear in mind that all bacteria doesn't have a nucleus. Instead, they have a simpler structure because of their small size, while the eukaryotics are the plants, the animals, the protista, which are sort of plants, sort of animals, but single-celled, and then we have the fungi. Next, so if we view the image of a prokaryote, you can quickly see how different they are from your typical eukaryotic cell. So if you remember from our discussion, right, we had so many uh, organelles and structures like cell membrane, lysosomes, vacuoles, vesicles, Golgi body, but the prokaryotes are much simpler than that. So if you can see, it's pretty much empty. Like some students would describe it as empty because the typical eukaryotic cell are very busy locations, while here you have a very simple, almost hollow structure. So let's take a look at the different structures therein. So first of all, prokaryotes do not have a nucleus. Instead, they have this not an official structure, but something called a nucleoid. So a nucleoid is not an official structure, but simply the clump of DNA found inside the bacteria. So they still have DNA, they still have genetic material. It's just that it's not organized, or it's not compartmentalized like that in the eukaryotes. Next, can they produce proteins? Of course they do, because they still have ribosomes. So the protein production is still possible because there is genetic material and then ribosomes to actually do the job. Although their proteins are going to be much simpler compared to that of eukaryotes. Next, can they move around as single-celled organisms? Yes, they can, because they have flagella. So their flagella are also simpler in structure compared to that present in the eukaryotic cells. How about their outer covering? Do they have cell membrane? Yes, the cell membrane is present for all types of cells. But aside from that, they also have additional covering, namely the cell wall. So remember, I mentioned earlier that plant cells have a cell wall. That is actually one of the characteristics that is common between prokaryotes, or the bacteria, and the plants. They have a cell wall. Furthermore, aside from the cell wall, they have a capsule. So they have three layers as their outer membrane. You have the plasma membrane, the cell wall, and the capsule. So the capsule is an extra thick covering which actually helps bacteria to survive in very diverse locations. So, diba, we mentioned that bacteria are everywhere. They are present in surfaces, inside our bodies, sometimes in the food that we eat. So how could they survive such different environments? Well, that's because of their covering. They have plasma membrane, cell wall, and capsule, which helps them maintain their internal environment very well. Alright, so they also have, of course, the cytoplasm, the collective term for all the materials inside the cell. So basically, when it comes to prokaryotes, all their cellular processes happens in the cytoplasm. They don't have like the the Golgi body, the vesicles, which are very organized in terms of their specific functions. For the cytoplasm, that's the location for all cellular processes. And since they're single-celled and quite small, they're fine with this arrangement. Alright, they also have a unique structure called the pilus. So we will be discussing that in a bit. There is a specific slide for that. Okay, so looking back at what a eukaryotic cell would look like, the eukaryotic cells are quite sophisticated in terms of, or quite complex is a better term, in terms of their structure. So one similarity they hold is that they have plasma membrane, which still serves as a selectively permeable barrier. So it still controls what materials goes in and out of the cell. Furthermore, they have a cell wall which protects them, like in plant cells, and they have a nucleus. So genetic material. Sorry, they have a genetic material. So the genetic material for eukaryotes are inside the nucleus, while for the prokaryotes, they're just called the nucleoid. Okay, so next slide. Okay, so how is this possible? Why is it that in the other hand you have eukaryotic cells that are very complex, multicellular, while on the other side you have cells that are very small and very simple? Well, to answer this question, you have to go back in the beginning of life here on our planet. So, touching on a bit of earth science. So, if you remember from your earth science discussion, the planet itself during its conception was incredibly hot, incredibly molten surface. But as soon as the Earth's surface cooled down, there are already remnants of life, and we have found fossils of this. And these fossils were left behind from by prokaryotic cells. So, safe to say that prokaryotic cells are actually the first organisms here on our planet. That's why they're so simple, that's why they're so small. They maintain that characteristic from billions of years, since the beginning of life up until the present day. So it's just that life started to evolve, life started to change. So as you can see here in our Earth's biogeological clock, the first ever organisms were prokaryotes, and then they evolved characteristics that enable them to photosynthesize. And here comes the eukaryotes. Since the cell has more capacity to produce energy, they can afford to be more complex and evolve better structures. So eukaryotes comes the algal kingdom, the invertebrates, the plants, the mammals, and then finally us in a very short period of time here on our planet. So, given that cells of prokaryotes are quite simple, it's easy to say that their DNA is actually quite shorter than that of eukaryotes. So, for example, the DNA of bacteria only ranges from 600,000 base pairs to 8 million base pairs. While for eukaryotes, the minimum is 12 million base pairs. So safe to say, the more complex the organism is, the more DNA is needed to actually give instruction to produce them. Okay, so here are more similarities and differences between prokaryotes and eukaryotes. So cytoplasm, both have cytoplasm, but eukaryotes have organelles, they have cytoskeletons, and their ribosomes are much bigger in size. For cellular reproduction, eukaryotes can divide through mitosis and meiosis, which allows further evolution since it provides genetic variability, while prokaryotes divide by simple fission. So if you're not familiar with the term, fission simply means divide. That's it pretty much. So you have one bacterial cell, it divides into two, that's it. The resulting daughter cells are completely identical to that of the parent. So not much events there. Next, you have the locomotion. So eukaryotes and prokaryotes both have flagella. But again, prokaryotes have simpler flagella in size, which I will be showing later. Okay, so I'm just keeping this. Alright. So I mentioned or pointed out a special structure only present in prokaryotes. So take note, this structure is only present in prokaryotes. They're not present in plants, animals, or whatnot. So the pilus or the pilus, depending on your preference, is a special characteristic of prokaryotes that enables them to share DNA through a process called conjugation. So this is not present for all bacterial cells, but are good to note since it enables them to evolve. So as we read from bullet 2, some bacteria are capable of conjugation in which a piece of DNA is passed to another cell. Now this is a very important characteristic because they can share DNA. Who on earth are actually capable of that, like directly sharing DNA? So, for example, two different types of bacteria can share DNA even if they're not the same species. So, for example, it's like um, making a dog share their DNA with a cat and it will be successful. It's that different in terms of prokaryotes versus eukaryotes. Now, why is this important? Because it allows them to evolve better. So, for example, if you're familiar with the concept of antibiotic resistance. So, antibiotic resistance is an ability of prokaryotes to survive against present antibiotics. So, when we say that disinfectants kill 99.9% of bacteria in any surface, then what happens to the point 1? The point 1 survives and shares their DNA, their ability to survive, to other bacteria, which actually creates a medical crisis in the present day because there are already strains of bacteria that cannot be killed by modern antibiotics. For example, there are cases of tuberculosis with they're called superbacteria, wherein antibiotics doesn't really work anymore. So this is dangerous because bacteria are again capable of conjugation, so they could spread their antibiotic resistance to other bacteria as well. Okay, so small but terrible here, based on our discussion. Okay, so in comparison again of the locomotion between prokaryotes and eukaryotes, I would like to emphasize that flagella is both present in prokaryotes and eukaryotes, but cilia is only unique for eukaryotes. So the flagella of prokaryotes, of course, they're smaller, so they would only require like a few strands of protein, and that would already enable them to move around the environment. Since prokaryotes are all single-celled, that would be enough for them to move around. In comparison to the eukaryotes, eukaryotes would need microtubules, multiple microtubules, in order to create one flagella, like here in the sperm cell shown in the picture. All right, so why is studying about prokaryotes actually important? Aside from they cause diseases and they are affecting our health and so on, prokaryotes are a key to understanding how life in our planet have evolved. So it's a point of interest in many scientists and of course science enthusiasts on what is the first ever living life form here on our planet. So based on the theory of evolution, that first ever organism is the ancestor of all life forms here. So that is termed as Luca, the last universal common ancestor. They are the precursor, the first ever organisms which resulted to the evolution of more organisms in our planet. So, based on the hypothesis, the Luca is most likely a prokaryote since we know that they have been present ever since the beginning of our planet. They are anaerobic. So, when we say anaerobic, it means they don't need oxygen. Take note that the early Earth is not rich in oxygen since there are no prokaryotic plants and algae yet. So there is less oxygen on the surface, but the prokaryotes can survive this as well as long as they're anaerobic. Furthermore, the early or the hypothetical Luca is heterotrophic, meaning they take nutrients from their environment in order to survive. Now, to divide all life forms here on our planet into three groups, we have the bacteria, the archaea, and the eukarya. So the archaea is a special type of bacteria. Their full term or their full name is archaebacteria. In terms of structure, they're still small, they're quite simple, but they're not like the bacteria because they are way special. They are way special in terms of structure. So let me show you. Okay, so when we talk about the archaebacteria, they are evolutionary related species that live in extremely inhospitable environments, and they are referred to as the extremophiles. So you take a bacteria and you give them special characteristics to survive extreme environments. That's your archaebacteria, and they are so special that they have to be placed in their own domain. So there are multiple locations here on our planet that we thought could not house any form of life because it's too harsh. For example, I was still alive when I was reading some books. I was reading some books about how the Dead Sea is called the Dead Sea because we thought that no life form could survive such level of salt in their bodies. It turns out there are actually bacteria very comfortable in it, and that's the Archaebacteria. So those bacteria are called the halophiles because they love such salty environments. More notable examples of extremophiles are the methanogens. So they use carbon dioxide in order to produce methane, which is another toxic gas. Acidophiles, they love acidic environments. They can survive up until to pH low as 0, which is already very deadly for other cells. For example, the acid level of tomatoes is 5. That's already acidic. What more kung 0? Next, you have the thermophiles, which live at very high temperatures. And stepping it up further, you have the hyperthermophiles, which can survive past boiling point. So you can try to boil them, but they will still survive. So they are often found in hydrothermal vents, which are the volcanic locations underneath the ocean. Okay, so to give you some examples, we have found organisms of the Archaebacteria domain living in boiling geysers like this in the Yellowstone Park. And the D. radiodurans is an Archaebacteria which can survive radiation from outer space. They don't need special protection, they don't need covering, they can survive radiation. So that's it's like the tardigrade, if you're familiar with that organism, they can survive very extreme environments. Um, that's why they're called Archaebacteria. So you might be wondering, then why are they not common? If they are better or they are more resilient than the normal bacteria, why are they not more prominent? Why don't we see them more often? Why are they less talked about in the scientific community? Well, that's because the Archaebacteria can survive extreme environments, but they cannot survive in normal environments. So, for example, if you take a hyperthermophile and put it in room temperature, it will quickly die. They don't like normal temperature. So, basically, they are confined in the current environment where they are in. Especially since these are extreme environments, they don't really spread out that much. That's why bacteria, the normal bacteria, are more common than archaebacteria. Okay, so not to, but now, aside from the archaebacteria, the main bacteria is present pretty much everywhere. We have found fossils underneath the Earth's surface, so several kilometers underneath the Earth's surface, and that means they're old. So we have fossils, which are their fingerprints, shall we say, from the early Earth, and then we also find them in other locations that you might not expect. For example, in the North Pole, in the South Pole. Here you have the panda. Sorry. You have the polar bears, which are green in color because cyanobacteria are living on the polar bears in a symbiotic relationship. It doesn't harm the bears for the bacteria to live there. It's just that the bacteria cannot live on the ice, they cannot live on the water, there are no plants, so they live on the animals instead. Okay, so how much do we know about prokaryotes as of now? We currently know that there are 6,000 species of prokaryotes identified, but scientists estimate that this is only 1/10 of the millions of prokaryotes that are living here on our planet. they are quite difficult to study, especially they are very small and they can be quite diverse, and it's quite difficult to identify them physically. For you to successfully identify a bacteria, since sometimes they look the same, it's better to use DNA sequencing. So this is a method to rapidly and cost efficiently identify microorganisms. So currently, we have collective genomes called the metagenomes, wherein we upload the DNA sequences of all living things, including bacteria. By that, we can compare the different bacteria by their DNA and identify if they are new species or not. Another venue for further studies are the microbiomes of humans. So, since bacteria plays a large role in keeping us healthy, for example, eating yogurts, drinking Yakult for lactobacilli, they do help maintain our stomachs, our digestion. So, for example, if you took antibiotics, after a while you might feel like you always get diarrhea. That means your internal good bacteria has been damaged by the antibiotics. So it's always recommended to eat yogurts in order to replenish your human microbiome. So not much is studied about this. There are some notable studies. For example, there is a study on mice wherein they took the bacteria of a thin mice to an obese mice, and then they found out that the obese mice actually lost weight because of the bacteria. So we're not sure on on why this is, if this is just a coincidence, but it is worth studying. So if ever you are interested in bacteria, microbiology might be a viable option for you in college. Alright, so this video is quite long, so I will be pausing here for the part 1 of our module 2. If you have any questions, feel free to message me in MS Teams or bring it up during our class session. Goodbye everyone, take care.